Hello. Oh, I don't have my video on. Just a minute. There we go. Hello. Let me remind you that today I am going to be having the lab at, well, the lab can be any time. I'm going to pre-record it. I've set the time for 5 o'clock, but you will need to go online to view this, and you can view it at any time. If you have any questions, I will not be here or in the lab to answer questions, so you'll need to email me your questions. So let's begin. Hopefully the volume is okay. That is a problem when there aren't students here. I don't have any way to know that. Let me see what the volume is here. That looks pretty good. All right, let's go to the syllabus. And you can see what we're doing in the lab today is Lab 4 on stains. And that's all we're going to talk about in the lab. So Lab 4 is on staining techniques. We're going to look at a simple stain, a differential stain, and special stains. You need to read the textbook. You should read about bacterial size, shape, and arrangement of cells from page 73 to 75. You should read about the bacterial cell walls, page 80 to 83. Simple stains, starting on page 65. Differential stains, page 65 to 67, specifically the Gram stain and the acid-fast stain. And then you need to read about special stains, page 67 to 68. There are 3 special stains we're going to talk about. The negative staining for capsules. And you should realize that a capsule stain doesn't have to be called a special stain. Depending on the stain, there are different capsule stains. Sometimes a capsule stain can be a differential stain, and other times it can be, well, a special stain. A special stain is a stain that we use to stain special characteristics of cells. And an endospore stain would be a good example of that. It's a special stain designed to stain the endospores that can be in certain bacteria. Another better example, even better example, because the endospore stain is another one that can be a special stain, but it can also be a differential stain, and we're not going to get into that discussion. But the flagella stain is always a special stain, and this is a special stain that we use to make the flagella of bacteria show up. Normally the flagella are so thin and hair-like structures that when we see them under the microscope, even if the cells are stained, we cannot see the flagella. And there are procedures you can do called the flagella stains that allow you to view the flagella. So you need to read about that. There will be video clips in this lab, and I think a virtual lab. I'll have to double check on that, but we'll go through, and then I'll point to you the virtual lab if it works. And I'm sorry, I just don't remember, which is really bad, because if we have a virtual lab, I'm the one who put it in here. So it occurred to me that I'm not positive that this is the latest version of the lab manual. So let me look briefly online just to make sure that I have the latest version. It doesn't really matter if I'm talking about the latest version or not, because I'm just trying to give you the major points. But on the virtual lab, it will only be there if we have the latest version. So let me look at that. Spring 2020. Ah, it looks good. Okay. So for successfully completing this laboratory, students should be able to prepare bacterial smears for staining, understand the principles and procedures used when performing a simple stain, a differential stain, and special staining techniques. Be able to interpret the results of a simple stain, a differential stain, and a special stain. And then be able to define these following terms. What's the simple stain? A differential stain, a special stain, what's a coccus, a bacillus, a spirillum, what's peptidoglycan, what are gram-positive cells, what are gram-negative cells, and you should at this point know some differences between gram-positive and gram-negative. What's an acid-fast stain? What is the primary stain? What's a mordant? What is the decolorizing agent, sometimes called the decolorizer? What is a counter stain? What is a capsule? What is an endospore? And what is a flagella? Because of the extremely small size, bacteria, when we look at them under the microscope, they are almost transparent. Therefore, it's necessary to stain them before you can observe them and really see them well. You do need to make a bacterial smear generally for all staining procedures. And how you make a bacterial smear is you simply put a drop of sterile water on a clean slide. Let me get that out of the way. And then add the bacteria, mixing the bacteria, and generally you only use a little bit of bacteria. You then smear the bacteria all over the surface of the slide, allow the slide to air dry, and then heat fix the slide. You need to do these last two steps, otherwise the bacteria cells will not stick to the glass slide. And when you're staining them, especially if you have a wash step to remove the stains, the wash step will remove the bacteria cells if you don't air dry the slide and then heat fix. A simple staining technique is where you use one and only one basic, actually I should say one and only one dye to stain the cells. Most of the time we'll be using a basic dye, but actually a negative dye is still a simple stain. And I don't know why I haven't corrected that. That was something our lead instructor made. And usually we use a basic stain, so most of the stains we use are basic stains. And we're not even going to talk about what the difference between a basic stain and a negative stain is at this point. But the point part is when you're doing a simple stain, you're only using one dye. A differential staining technique usually uses two or more dyes, and it provides additional information about the cells. Okay? In a simple stain, all the cells will be stained the same. In a differential stain, the cells will be stained at least two different ways. And we'll see that with the gram stain, which is the most common differential stain. And then the other stains we need to talk about are special stains, and they're used to stain special structures of the cells, like the flagella or the capsule. All right, in this picture we're looking at different cells, and these cells are cocci. And you can notice that the cocci can grow in different cellular growth arrangements. They can be single cocci, which aren't shown here. That would just be one cocci. And cocci are ball-shaped cells. They can be diplococci, which is shown here. They can be tetrads, shown here. And I have no idea why this is not in the right place. Of course, it's not right anyway, so I'll just keep going as I'm going. This right here is staphylococci, and then this right here is streptococci. So streptococci is when the cocci are in a chain, and then staphylococci literally means cocci growing in a grape cluster, because it looks sort of like a cluster of grapes. All right, so those are the common cellular growth characteristics of cocci. There are a few others, like Sarracenia is an 8 cellular arrangement, so it's 2 cocci together, stuck together, and we call that Sarracenia. The important point is that the cocci have the most cellular growth arrangements. The bacilli have fewer growth arrangements. You can have single bacilli like this. You can have diplobacilli, which isn't shown here, but it would be like that except bacilli instead of cocci. And you can have streptobacilli, which isn't shown. And let me think, what's another one you can have? Oh, there's another one, it's not shown here, so I'll talk about it later. And then this picture is showing you there are some other shapes of bacteria that are not really a cocci and a bacilli. The first we need to talk about is a coccobacilli, which is, I guess, that. And it's sort of a very, very short bacilli that's rounded, and so it looks similar to a coccus, but if you look at it, I'll try and blow that up. I think I do it right here. If you look at it, especially with that cell, you can see that's not really a coccus, and it's not a bacilli, and so we call those coccobacilli. It's in between a coccus and a bacillus. Hopefully you have no questions about that. And then there can be pleomorphic cells, and that's where the cells have different shapes. We'll talk more about that later in the term. This is not actually a very good picture of that, but you can see that that cell looks different than this cell here. And then there are spirilla, and I don't like this term spirilla bacilli, because these are spirilla, they are not bacilli. And so I'm just going to call them spirilla. And those are cells which have a curve to them, usually an S curve, like this one here, and that one's a gentle S. That one, I don't know if that's an S or not. It might be just more of a, what do you call that, convex shape. And you can also have multiple curves, where it curves multiple times, and I'll try and draw that here for you, where it curves. That's not really working too well. Hopefully you can see my mouse, where it bends like this and this and this and this and this and this and this, and that is called the spirochete. We'll see a picture of that later. Let me shrink the size of this. Right there, I think. So the differential stains, the main one is the Gram stain. And what's the other one? The acid-fast stain. The Gram stain was invented in 1884 by Hans Christian Gram. And so whenever you spell the Gram stain, you should be using a capital G, because it's named after a person. And so we go the capital G because Gram was a person. And he invented this stain so that he could distinguish between the patient's cells and the cells that were infecting the patient. And then microbiologists started using the stain that he invented, and they said, my God, this does more than differentiate between a patient's cells and the bacteria cells which are infecting the patient. This stain can differentiate between bacteria. And so this is the most commonly used differential stain in microbiology, because with this stain we can separate almost all cells into gram-positive cells, which stain purple, shown here, and gram-negative cells, which stain pink, shown here and here. And I don't know what happened to this picture. It moved or something. Let's see if I can move it over. Oh, that's a little better. And so the gram stain allows us to differentiate almost all bacteria into two groups, and that is why it is the most commonly used differential stain in microbiology. And here we're looking at the gram stain, and as you can see, there's many different cells here, some that are gram-positive. I think that's a gram-positive coccus, and then gram-negative, those are gram-negative spirilla, and there's a gram-negative bacillus. If you don't know, the gram-positive are purple, the gram-negative are pinkish or reddish. The gram reaction is based on the ability of the bacterial cell walls to retain the first dye of the gram stain, which is crystal violet. Gram-positive microorganisms contain a thick peptidoglycan layer, shown here, gram-positive cell, and that thick peptidoglycan layer traps the gram stain and the iodine, which I'll talk about briefly in a few minutes. It traps it and doesn't allow it to wash out of the cell wall in the decolorizing step. On the other hand, the gram-negative cells have a much thinner layer of peptidoglycan, and they also have a lipid outer membrane to the cell wall. When the crystal violet and iodine first stain these gram-negative cells, they do stain purple, but when we decolorize the cells, the crystal violet and the iodine washes out of these cells. And it's just because of the thickness of the peptidoglycan layer. All right, after the bacteria cells are stained with crystal violet, we then use Gram's iodine, and you should know that that is a mordant. It is something that complexes with the crystal violet, making a larger molecule. And then we do the decolorizing step, and then we counterstain with safranin, which will stain the gram-negative cells a pinkish color. All right, hopefully you have no questions. If you do, email me. All right, some gram-positive bacteria lose their ability to retain crystal violet dye and are easily decolorized. Therefore, we describe these species as gram variable, and they will appear as a mixture of gram-positive and gram-negative bacteria having the same shape, like this right here, all has rods. And if you look very carefully at these cells, you will note a difference, that the gram-positive cells have distinct boundaries, the cells are very sharp, and the gram-negative cells, let me blow this up, their boundaries and the cells are not very distinct. Like this one right here, it almost looks like a ghost cell. And that one right there is not as distinct as either of these two. And this one right here, it looks like the cell is fading away. And often on the cells which we call Gram variable, what happens is that the dead cells are staining Gram negative, which is why these are stained so poorly and they're not sharp. And then the cells which are viable stain Gram positive. And that's to be expected. If the cell is dying, it may not retain the crystal violet. Anyways, the most commonly found bacteria that are called Gram variable are the Mycobacterium. And if you use the Gram stain correctly, the Mycobacterium will usually stain Gram positively. But if you use old cells, the Mycobacterium will stain Gram variable, because the dead cells will show up as Gram negatives, and the viable cells will show up as Gram positives. And whenever I've had a student who had Gram variable results, I always ask them, how old were your cells? And they would say, oh, these cells were pretty old, like 2 weeks old. And I said, you can't use old cells for the Gram stain. Otherwise, the Gram stain may not work correctly. And the reason why you're getting this Gram variable result is the Gram stain should only be using freshly grown cells. For you guys, freshly grown cells mean that they've grown for 2 days or less. If anything beyond 2 days, they're not freshly grown. And you should not use the Gram stain because it may not work correctly if you don't have freshly grown cells. And that's actually true for all of the Mycobacteria I've ever looked at. Admittedly, I've only looked at 2 or 3 species of Mycobacterium, but of all the species I've looked at, they always stain correctly if you use freshly grown cells. So here's some tips for preventing Gram variable bacteria. Avoid using old cells. Prepare very thin smears. If you have a very thick smear, the stain which is supposed to get inside the cell may not reach the bottom cells because it's too many layers of cells to get through. And conversely, if the dye is supposed to wash out of the cells, if the layer is too thick, not all of the stain may wash out of the cells. Heat fix the bacteria only after the smear has been air dried. If you don't do that, you cause the water to evaporate in the cells, and that destroys the cell wall. And so the staining won't be correct if you do that. Avoid overheating the cells during heat fixation. And if you overheat the cells will literally start to melt. And of course that will destroy the cell walls as well. Use fresh staining reagents. Do not over decolorize, and do not under decolorize. And that's a tricky step. Okay? And if you don't have a slide that's prepared well, look at the cells where they're thin and evenly dispersed. All right? Hopefully there's no question about the Gram stain. Let's move on to another differential stain. That is the acid-fast stain. Some bacteria, particularly Mycobacterium and Nocardia, have a waxy substance that we call mycolic acid in their cell wall. These bacteria are able to resist acid-alcohol decolorization. and we therefore refer to them as acid-fast bacteria. The acid-fast cells have so much waxy mycolic acid in their cell walls that they do not readily take up dyes that are water soluble, but they do take up acid fast or acid fast dyes, and then they retain that acid fast in their cell walls. And in the acid fast stain, the acid fast negative bacteria stain greenish in the stain, but the stain could also be bluish depending on what your counter stain is. And then the acid fast bacteria, the acid fast positive bacteria, stain pinkish or reddish. Okay? And so here we might have a person who has Streptococcus pneumonia, and we ask them to make a bacterial sputum, and they then stained it with the acid fast stain, and you can see the Mycoplasma pneumonia staining pink. And then other bacteria, like from the mouth and the throat, will stain greenish. All right, let's move on to the special stains. Remember, there are three we're going to talk about. The first is a negative stain that we use to stain the capsule. And a capsule is the structure we can see under the microscope because it's thick, it has more molecules than just water in it, and you can see it under the light microscope. But the capsule is part of the glycocalyx or glycocalyx, both pronunciations are correct, and that is a structure outside the cell wall that the cell secretes. And if you can see it, it's a capsule. And so that's what the negative stain is, to determine if the cell has a capsule. Now, with the light microscope, you could not see a glycocalyx, which is not a capsule, like a slime layer. It is a glycocalyx, but you would need an electron microscope to actually see this glycocalyx. Okay? Because it's mostly water, it just does not show up with the light microscope. The productions of capsules is important because it's associated with an organism's virulence or its pathogenicity. So many of the bacteria that produce capsules are pathogens, human pathogens, meaning they're virulent. And you can read about this, telling you about some of them. The capsule also gives the bacteria, when you look at it on a petri dish, it gives the colonies a very mucus-like appearance. And that's what this is here. Streptococcus pneumoniae with the capsule. When you stain the cells with India ink or nigrosin, that would be a negative stain, and the background of the cells will stain. And with nigrosin, and this might have another stain in there too, the cells will stain. With India ink, the background is stained very darkly, and then the cells are stained very lightly. So this says it's India ink, but I can tell by looking at this picture is not India ink. And then the capsule does not stain, and that allows us to visualize the capsule. Okay? So that's why we use the negative stain. It stains the background of the cell, and then it also stains the cells, but not the capsule. Another special stain is the endospore stain. Endospores are metabolically inactive cells, and they're very hardy structures that are produced by some bacteria, and they're a defensive strategy so that when the environment deteriorates, causing the parent cell to die, the parent cell realizes the environment is deteriorating, and then the cell can make an endospore, and then this endospore is a cell in hibernation. And so the cell can survive even when the environment is very unfavorable to the cell. Like I said, endospores, the cells are metabolically very inactive. And then the cell just can survive. And it can be for years, decades. They have a record. I'm trying to remember. It's either something like 400,000 years they've obtained, or maybe it's 400 million years, something like that. They've obtained endospores from ice cores in Antarctica and then thawed them out, and the endospores made it a favorable environment. The endospores then started germinating and the bacteria started growing. And like I said, some of these endospores can survive for a very long time. And I don't remember the exact record, but it's something like 400,000 years, or maybe it's 400 million years, and the cell was still alive as an endospore. Most endospores don't live that long, but there are a number of them that they've isolated, like from the Egyptian mummies, and they've been in the mummies for something like 8,000 years. And so endospores can survive for a long period of time. Now remember that an endospore is a structure the cell makes for survival. It is not a reproductive structure, as one parent cell makes one endospore. That is not reproduction. And then the parent cell dies. The cells which tend to make endospores tend to be in two families, the family of Bacillus and the family of Clostridium. And this table shows you some of the common endospore forming bacteria. All right, the last special stain I want to talk about is the flagella stain. Flagella are long thread-like appendages, sort of like a whip, that are attached to the cell surface, and their purpose is providing the cell with motility. So if the bacteria can swim through the fluid in a liquid culture, that means they have flagella, because the only way bacteria can swim is with flagella. Now with eukaryote cells, they can swim by flagella, but they can also swim by cilia, which bacteria do not have. And then eukaryotes can also move by pseudopods, which bacteria do not have. Anyways, the purpose of the special stain is that what it tends to do is it coats the flagella many times, and so instead of having just one flagella, you have, whoops, not showing it, you have many layers on top of layers. And then when we look at the cells, you can actually see the flagella. And then this table, you'll need it to answer one of the questions in the worksheet. It's giving you the number and flagella distribution of the bacteria. And you don't really need to know this except for the worksheet. Meaning I'm not going to quiz you on, this is Peritrichus, and this is Amphitrichus, and this is Lophitrichus, and that's Monotrichus. whenever you see a specimen like this slide here, or this picture here, and you want to determine the flagella arrangement, you take it on the cell that has the most flagella. Like this cell here, I only see like 2 flagella on it, maybe 3 if that's 2 right there. And this one here has a couple of flagella, but it's nowhere near like this cell. And so you go with the cell that has the most flagella. So this is all one species of bacteria, and you'd say that the flagella arrangement is the highest order, which in this case would be Peritrichus. So if you have any questions about that, email me or watch this video because I just explained it to you. All right, you should know the terms of this lab. Sorry, I had an itch there. You don't need to click on the references. And I think that's it for the lab. Let me scroll down. Up. All right, the laboratory exercises. You're not physically going to be performing any of these activities, but you must understand how to prepare bacterial smears for staining the cells, the principles and procedures used for making a bacterial smear, the principles and procedures used and the various staining techniques, and be able to interpret the results when you look at the slides that you stain under the microscope. In procedure 1, you need to know or learn how to prepare a smear for bacterial staining. And what a smear is when you put the bacteria on the glass slide. And we have a little video clip here for making a bacterial smear, and then you air dry and heat fix the slide, and we have a little video for how you do that. So watch those. And let me state, since you're not here, that students, the most common problem they make when they make a bacterial smear is they use too many cells. You want to have only a little bit of cells. And what I'm talking about a little bit, when you have a loop, you don't have the loop full of cells. You have like 1/8 of the loop full or less. And then you smear that over the slide, and the cells should look slightly milky. If they look milky, like you put milk on the slide, you've got too many cells. So they should look slightly milky. And then go through the procedure for preparing and smears and staining. It's here. And once you have made the bacterial smear, you can then stain it. And the first procedure we'll do, procedure 2, is doing a simple stain. And so watch the video clip on performing a simple stain. Remember, you're only using one dye in a simple stain, and it stains all the cells the same, depending on what the color of the dye is. Usually you use methylene blue or crystal violet, but you can use other dyes for making a simple stain. All right, read through the procedure, and here we have a picture of cells that were stained. E. coli over here, and what is that, Staphylococcus aureus here, and you notice they're all staining purple. Let me shrink that down. This is a little too big. The cells are looking weird. There we go. Close enough. And that's because the cells were stained with a simple stain. They all look the same. And then on the worksheet right here, describe the cellular morphology of E. coli and Staphylococcus aureus. I should remind you, please do not put your answers in the lab manual. It's too big and it takes up too much room on Canvas. Please put your answers in the worksheet and then submit the worksheet. And I've got the worksheet right here. So let me pull up the worksheet. And like I said, it has the same thing here, so I probably should be going through this here. And then put your answers in here. And then when you're all done, submit the worksheet. Alright, the next procedure, procedure 3, is on the gram stain. Watch the video on performing the gram stain. And you can practice this. Let me make sure this works. On the virtual lab. Ugh. That was rather loud for me. I just want to make sure the virtual lab works. Of course, I should do it from Canvas, not from here. No, that one's not working. Let me look here. I apologize for not remembering. If we have a virtual lab for this lab or not. Let's try here. Yeah, this one's working.
Hi, Lab Assistant. Welcome to the yogurt section of the dairy processing plant.
Okay.
It appears that our latest batch of yogurt might have been...
Alright, so the virtual lab for the gram stain does work, at least from Canvas. I need to update my own version, and I was wondering about that. Let me double check to make sure the virtual lab is working in the worksheet. Too far. There we go. If it doesn't work in the worksheet, I'll get it working, but Alright, so in the worksheet you don't have any links. You'll have to get the links from the lab manual. But the link is working on Canvas, so please watch the virtual lab. The virtual labs are really the only way you're going to get any experience for doing the techniques of microbiology. And we're looking for more virtual labs, and we had a number of them, but they all use Flash. And if you don't know, whoever made Flash, I don't remember, maybe it was Adobe, they stopped allowing you to use Flash. And so we only have a few virtual labs, but I found 2 or 3 for this term. So that was an improvement, trust me. All right, so let's go back to the worksheet. Anyways, go through how you make the gram stain, and then answer the questions, the smears, the E. coli. Tell me if it's a gram positive or a gram negative cell. And the same with the Staphylococcus aureus. Is it gram positive or gram negative? And then tell me the cellular morphology, if they're coccus or bacillus, the cells. And then answer these questions. Each of the following organisms, predict the color you would see. Pink, purple, or colorless after the gram staining. and then do procedure 4 on the acid-fast stain. There's a video clip to watch on that. And then answer the questions. And then there's procedure 5, the special stain. We have an endospore stain, a negative stain, and a flagella stain. We have videos on the endospore stain and the negative stain, so please watch those videos. And then here are 3 bacterial smears being observed. And then answer these questions, which are in regard to the 3 bacterial stains in the pictures, 3 pictures above. All right, if you have any questions, whoops, I'm not quite done. And then there's 2 case studies for this lab. Case study 1, read through it, and you're looking at the x-ray pictures of a patient and a bacterial sputum smear, and then the Nielsen stain. And you have to look that up, what that's staining for. And you can see green cells and red cells, and that means something. And then tell me, what is the most probable diagnosis? And you should tell me the species that is causing the bacterial infection. You can tell me the disease as long as you're specific on which disease it is. But I prefer the bacterial species causing the infection. And then we have a case study 2. Here's the patient. Obviously had a bacterial infection in his foot. And then there's the information about the patient. They did a gram stain on the patient's cells in the foot, and we saw a gram stain here. You can tell me if that's gram positive or gram negative. And then they did the Schaefer-Fulton stain on these cells here, and we can see special structures in the Schaefer-Fulton stain. So tell me what that structure is, and then look up in the table here. You need to use this table up here. Lost it. Here it is. For telling me what species is likely to cause that infection. You can tell from the studies. Okay? All right. And that one I'm going to require you tell a species, because if you just say it's a bacterial infection, that is not enough. Okay? All right. If you have any questions, please email me, and please put your answers in the worksheet. Remember that we do have a virtual lab, which is in the lab manual for today. All right. Good luck, and I will see you at 6:30 for the lecture today.